The main findings at the moment is just that regulatory T cells are involved in suppressing this type of T cell priming event, and that this is specific to the pulmonary draining lymph nodes of the lung tumors, because in our study, for comparison, we're injecting the same cancer cells into 2 different locations in a mouse, and so we're comparing how the immune responses in the lung are different to immune responses in the subcutaneous space. And in making this comparison, we have found that the suppression phenotype that we're seeing is specific to the lung setting, which is very interesting because it makes us think about how different tissues might induce different immune responses. And I don't think we've really thought about immune responses in this way before. So yeah, I think that's probably the lung specificity of it is another interesting phenomenon, and we're still trying to work out why exactly this is happening in the lung. But now that we know the molecular interactions with Tregs are important, I think we'll get there soon. Brendan's first observation was actually that the lung tumors do not respond to checkpoint blockade immunotherapy while the subcutaneous tumors do. And so this whole project really started to understand why and how. And the reason this is valuable is because there is a large number of patients with non-small cell lung cancer that have T cell infiltration but do not respond to checkpoint blockade immunotherapy. And so we're trying to understand these additional mechanisms of suppression that might be very different and that might require a different therapeutic approach. And so we think that understanding these mechanisms could lead us to that.